can we just look at it here? See if you can see it here. Because you're toward the end here. Which one is it? Do you know?
It would be this guy.
Yeah, I realigned everything.
Okay, thank you. Then
That should be good. Yeah, because the formatting from PC to Mac like screwed everything up. So I was up fixing stuff. Okay, so for everyone who's here, thank you for being on time. We're going to give it the UCLA a couple extra minutes.
Do we get a name for being on time?
This is a pass-fail class. There's no grades.
I'm sorry to hear that.
You pass. So we'll start in a couple minutes. Okay. So, good afternoon, everyone. I see a lot of faces I recognize and some that I don't, so that's good news. For those of you who don't know who I am, my name is Adam Stieg. I'm one of the associate directors here at the California Nanosystems Institute at UCLA, and on behalf of CNSI Director Jeff Miller, who's in the back, and all of our colleagues from CNSI, I'd like to welcome you all to our open house. It's been a number of years. We were just chatting about when the last time we did this. It was certainly pre-COVID. I think it was at least 7 or 8 years ago. We used to do this every year to highlight all the new capabilities and exciting things that we're doing. And so we're really glad to kick this back off again, open our doors to the community. I know we have a lot of folks from UCLA. Who's from UCLA? All right, about half the hands. Who's not from UCLA? Okay, we got a bunch of hands that are not from UCLA. That's good, right? We serve the local, regional, and international community, and so it's exciting to have people here from different communities. And so what I'm going to do right now for all those folks who are not from UCLA is to give you a little bit of an intro into who we are and what we do here at the CNSI, and then I'm going to turn it over to the directors of our shared facilities. They're each going to give you a sort of highlight, quick 5-minute sizzle reel about of all the exciting things that they do. We're going to then take a chance to open up the labs and give you a couple hours to go and visit and ask questions and see what we're up to. And then toward the tail end, we're going to have a reception, drinks and food and snacks to network and connect and ask questions that you may have not had a chance to ask during the tours. And so for the next 4 hours, to the extent that you all want to be here with us, we're excited to be here with you. And so I'm just going to take a few minutes to give you a quick sense of where you actually are. And so the California Nanosystems Institute is a partnership operating under the University of California system. There are 4 what are known as the Institutes of Science and Innovation. They cover different domains of science and technology. We are partners with our colleagues at UC Santa Barbara in the area of nanoscience. And what we do here generally, although we're not an academic department providing degree programs for students, we educate and we bring together folks from disparate fields. We do team science initiatives to address grand challenge problems that are sort of facing society, and we do it with all deep domain expertise that each of us brings to bear. And so we are a sort of ecosystem of innovation. As part of the university, we also have a mission to not just produce papers in fancy journals, which is always great. Everyone likes a science and nature paper, but ultimately to get those technologies and the solutions out into the wild for the benefit of society. And so we also are the place, which you won't see here today, but the place where we incubate technologies, translate and commercialize technologies. And so on the 6th floor upstairs, we have actually 30 startup companies operating inside this building. So we develop and foster a culture of entrepreneurship amongst our faculty, our students, and our postdocs. And then, of course, even though we are not an academic department, we are training the next generation of scientists and engineers. And whether that's in traditional university classes, in laboratory, hands-on training opportunities, workforce development, some of these things, the CNSI really addresses all the critical areas of innovation, commercialization, and education here at UCLA. So without going into the gory details, what I would say is that pipeline going from left to right is really facilitated by our research infrastructure. And so at both CNSI sites on both campuses, there are large-scale investments in our research infrastructure. Our technology centers, which is what you're here to see today, are one of these foundational pillars that provides access to researchers to explore their exciting solutions. We then bring these teams of folks together and we form centers. So you see some examples in the center here of large-scale, multi-year, multidisciplinary, and multi-investigator centers that come out of and leverage the infrastructure and the sort of ecosystem of innovation that we have here. And then, of course, we translate those into examples where we're commercializing technology, a lot of which comes out of UCLA, and we're trying to push that forward out into society. And so this culture of innovation, right, leads to incubation, right? So we're not here to see Magnify, which is our startup incubator today, but for those of you who may be interested in the startup space or entrepreneurship, a lot of that is driven, again, by our technology centers. We're not a hotel for companies. Our companies are here for a reason. They're here because of the proximity they have to the investigators that they may or may not be collaborating with and taking advantage of our research infrastructure. And so we have a number of companies over the last decade that we've incubated that have been very successful in fundraising and creating new jobs and new markets in and around the Los Angeles region. And so that's the next step on that pipeline of what we do here at CNSI. And then, of course, we need to keep this flywheel going, right? And so we need to educate the next generation. So from K through 12 outreach into the local community in Los Angeles, training teachers, working with students, developing educational modules that can be deployed in sort of the youngest of the young generation, all the way up to workforce development and professional training programs that we are deploying in concert with our facilities and other partners throughout the region. We are engaged in non-traditional university education activities, but we do play a very critical role in that aspect. But what you all are here to see today are our technology centers. And so you may recognize technology centers more often described as user facilities or core labs. We call them technology centers because they are hubs where we are developing, pushing, and deploying advanced technology. We're not always providing standard stuff. We're building and designing and making things do what they weren't designed to do, specifically to meet the needs of the research community that we have. And so you see here, we have over 100 unique services distributed across the different facilities that you'll hear about today. We have somewhere between 1,500 and 2,000 unique researchers come here. And since we opened our doors about 15, 17 years ago, we've trained over 10,000 researchers to utilize the advanced capabilities in making and measuring screening of all sorts of things at nanoscale dimensions, materials, devices, living systems. And so the technology centers follow this pipeline, right? So we are open 24/7, 365 days a year. One of the reasons that a lot of the hands went up that were not from UCLA is because we're open to anybody. Academia, non-UC academia, industry, government labs with no preference and no priority, right? Everybody can come here and use these facilities on a fee-for-service basis. We provide access, infrastructure, expertise, and most importantly, training, right? We're not generally a service provider. We want to teach you all how to do these measurements while in collaboration with our team to be able to get you where you need to go. We take people that are novices and we make them experts. We allow experts and we work and collaborate with them to push the domains of knowledge forward through innovation and development. And so as you see here, we have a lot of different things that are going on. The labs are used quite heavily. If you were to stand at the front door and you looked at who's walking into the building, I would say probably between a third and a half of everyone who walks through the front door is coming to use the facilities here, in addition to all the other exciting things that happen within the Institute. And so on the lower left, you'll see this is just some high-level things that we'll highlight. I won't get into the details because you're going to hear it from my colleagues, but you see that part of what we do, as I said, is that we're not just providing standard services. We like this sort of feed-forward loop that involves obviously advanced instrumentation, but developing technology, providing training, providing service, collaborating, disseminating, and having that be a virtuous cycle where we continue to deliver real value to the community of science in Los Angeles. And so you're going to see 6 labs today. There are posters. I'm not going to go through this, but in the basement level, my sort of joke is that actually I was at the Getty Conservation Institute with some folks that were here yesterday. They have beautiful labs with giant windows overlooking the mountains. It makes me a little jealous. But what I always say is that our basement is prime real estate, right? If you don't want a view of the ocean, but you want a view of atoms and molecules and the smallest of the small, you need to be in a quiet, isolated place. And so the basement level is where we do our measuring. And so you'll see that there are 3 measurement suites, which are 3 main pillars: optical, electron, and scanning probe microscopy to characterize and measure the things that people are making, right? And as you move up, you will go to level A. For those of you who are interested in microelectronics, semiconductor manufacturing, we have our clean room. One of our 2 locations at UCLA is operating on the A level of the building. Don't worry if you want to go see the clean room, but you don't want to put the bunny suit on. I don't think you're taking people inside, right? No, so we're not getting gowned up. It'll just be a discussion, sort of window tour if you're interested in what we have there. On the second floor, you'll see an example of an automated synthetic biology platform that's designed for materials discovery, in particular in the space of sustainable biopolymers. And then all the way up on the 6th floor is our drug discovery and high-throughput screening facility, which is really, really unique and exciting. So all of these labs are going to be open for you. After we stop talking, you're going to have a chance to go up. We're not doing curated tours where we're signing up on sheets. We want it to be sort of flexible and free-flowing, right? So all of the staff for the labs are going to be here. We have a lot of people that support these labs, many of which are, PhD-trained folks. So you will see a lot of familiar faces throughout the day as you move through the laboratories, as well as the people who are going to talk to you next. But these are the people that you're available to talk to, right? And so the biggest thing that we do at the beginning is that we open our doors and we want people to come in, right? We want to talk about where your challenges are. We want to understand what you're trying to do and define whether or not there's a near-term opportunity to give you what you need. Or, which everyone has heard me say before, the answer is not no, right? Even if it's not necessarily something we can do now, our goal is through, calling ourselves developers of technology is to find solutions, to develop solutions, to make technologies that we have work in different ways to address the problems that the community brings to us. And so a lot of this starts from conversation. And so we're hopeful with our open house that you can come and see and ask questions, understand what's possible, and hopefully maybe define some future directions for you to come back in the future and utilize our resources. And so with that, I'm going to turn it over to our first speaker, who is Laurent Bentolila. He's the director of our Advanced Light Microscopy and Spectroscopy facility, which is our optics characterization facility down on the B level. And then we'll just go through a sequence. That sequence that I said, there's going to be 5 or 6 minute talks, 5 or 6 minute talks, gentlemen. Right? And then we'll do a quick wrap up and head out and get to show you all around the lab. So again, thank you for coming. I'll probably thank you again at the end, but appreciate your attention. Laurent, the stage is yours.
Thank you, Adam, for this wonderful introduction. My name is Laurent Bentolila. I'm the director of the Advanced Light Microscopy Spectroscopy here, and I'm going to do my best to tell you what we do, or at least a little bit of what we do in 5 minutes. But essentially what I would like you to remember is that using light and optical microscopy is very versatile, and we do imaging at all length scales. So from the nano to the microscale, usually using fluorescence microscopy, so we can do and characterize single molecules all the way to tumors in small animals. And how do we do that? Essentially, most of what we do is fluorescence-based measurements. We do that at all length scales, and we are using 6 dimensions in our imaging. So the XYZ, that's your 3-dimensional imaging, and we can do that from nanometer to centimeters. Temporal, we can image from nanoseconds to days, and spectral, we can do it from the UV all the way to the infrared. So here are some examples. Do I see my cursor somewhere? Yep. To illustrate how we can actually mix and match all of these dimensions, here is an example of 3D volumetric imaging. Most of the examples I'm going to use are taken from our biological samples, but we do a lot of nanomaterials as well. Is it playing? No? Too bad, because it's a fun movie. So here's another example of 3D imaging here combined with spectral, so multiple colors in one pass in 3 dimensions. Here we are looking at metastatic cells in green, actually covering a blood vessel in red, pretty deep into the brain of a mouse. So very powerful. You can slice it in every direction you want to really understand how this metastatic process develops over time. Here, we can use non-linear optics to probe even further deep into the tissue. Here, what's interesting is the pink signal is actually unlabeled structures into your sample. That was collagen. So we can do that by using a process called single harmonic generation. So here what I wanted to illustrate is the fact that we can image unstained samples through these kind of nonlinear processes. And here is a really gigantic slab of tissue that we can actually image in 3 dimensions, combining 2-photon microscopy and SAG. So 4 dimensions again. And here we can image really fast using 4 dimensions, X, Y, and Z, looking at a super fast volumetric imaging here of the beating heart of a live zebrafish. So from that kind of movie, you can understand that optical imaging is non-damaging, so we can image actually live samples in real time. All right, and another very good assets that we have is we have been one of the first labs here to introduce super resolution optical microscopy in order to close the gap between what a normal light microscope can do, which is limited by light diffraction. And this is what you will usually see if we want to relate the structure and the size of the structure we image with an electron microscopy image. So that would be your confocal microscope, about 200 nanometer resolution. Here we have, here at CNSI, and we'll have demos if you come to the lab, STED microscopy, where you can push the limit to about like 20 nanometers. That will be your little structure here in that electron microscopy image. And we have STORM, imaging that can push even the limit even further down to around 10 nanometers or less, and that will be that little structure in blue. And here are some examples. Again, here STEM microscopy is very powerful because you keep all the advantages of confocal microscopy. You can image live samples, you can do it in 3D, and you can do it in multiple colors using your spectral capabilities. And you can, of course, apply that to nanomaterials and biological samples. Here is our team. That's our expertise, so please talk to us. We are here. Ellie here is in the back, and Brian is probably in the lab, but please talk to us. And really, our expertise is not just the instrumentation, but our knowledge. We have a lot of experience. We can customize microscope for every experiment. We can assist you with sample prep. We consult to really design the entire pipeline of your imaging experiment. And we are also very well versed into data processing and visualization and customizing your experiments. So with that, it was really a bird view of what we do. So if you want to see more, please come downstairs and talk to us at any time. Thank you so much.
Okay, so our next speaker is just calling me right now. He's not here, so hopefully everything is okay. So we're going to jump ahead quickly past electron microscopy, and we will come back, and I'm going to turn it over to Paul Chang, and then I'm going to go answer my phone and see what's going on. So please, Paul.
Hello, everyone. I'm Paul. I'm working at Nano and Pico Characterization Lab, mainly located in the B139. After a quick review presentation for 5 minutes, if you are interested to learn more about us or to see all our equipment, please stop by in B139 during the lab tour sections. So at MPC, we specialize in surface analysis such as the size, shapes, height, and depth profile, roughness, as well as mechanical, electrical, magnetic property using the scanning probe microscopes. And also we have a size analysis and element analysis for those applications at the bottom of the center. We have 3 full-time staffs, the Adam, the director, and myself to provide the training and technical support, and Mike to focus on the bio-AFMs. So we offer the consulting of the design of the experiment or research, and also the training, in addition to also assistance with imaging or data acquisitions. So here is a snapshot of the all equipment we have at MPC. So we have 3 different type of the SPMs for different applications. And also we have a DLS and NTA for size distribution, surface charge, and counting number of the particle analysis. And ICP, OES, and MS for elemental analysis. And rheometer is for the mechanical property analysis, such as the viscosity, viscoelasticity, or shear stress. So I would like to introduce the 3 practical examples how we can support your research using the equipment at MPC. The first example is microelectronics. So here is some example of the sample, the single layer of the molybdenum disulfide. So by utilizing the SPM or AFM, you can analyze the shape of the sample, which shows the triangle shapes. Also measure the thickness of the thin film, showing around 7.5 angstroms. And not only that, also we can create the 3D images by using these AFMs. On top of this, another advantage of this AFM is we can analyze the different type of the characteristics, such as the mechanical or electrical magnetic property. In this case, it's a piezoelectricity. So by using this AFM, you can obtain the IV curve with applying different amount of the force. So you can see the IV curve is shifting. The slope is different depending on the force we are applying on the surface. So based on this application, we can measure the different electricity, which is a piezoelectricity, the property using these AFMs. Another advantage we want to highlight is biomechanics. So we have a bio-AFMs, which called this a nano wizard, integrated with the confocal microscopes. So this bio-AFM is specialized to take three different type of the images. The first one, number one, is optical images, confocal images, another scanning probe microscope images. By utilizing this one, we can see overview of the tissue sample with the optical microscope, and we can identify the different area of the tissue, which is this green is collagen from the tendon, and this dark pink is from the elastin from the bone tissue samples. So we can identify different area using the confocal microscope. Also, we can overlay or mapping the scanning probe microscope images to see the different topographic information about the size, height, and depth profile. And the beauty of this bio-AFM is we can add additional information such as the stiffness in this case. So you can see there are black line represent the different soft tissues between the junction of the tendon and the bone tissue samples. The last example we want to highlight is the drug delivery. So when you're working on the nanocarrier for chemo or immunogenic the drugs, you can use the DLS or NTA to analyze the size distribution, or surface charge to optimize your experimental condition or parameter to enhance your bioavailability or the stability of your nanocarriers. Not only that, also you can use the ICP OES or MS to track down drug releasing profile over the time in the different medium. Also analyze the different type of the tissue to understand how this drug is spread in the body, especially for the tumor site. This is an overview of the MPC, the tech centers. I understand you may have a lot of the question about the MPC, how we're doing. So please, again, stop by the B139 during the lab tour section, or send the email at mpc@cnsi.ucla.edu, or scan this QR code to get the more detailed information. Thank you for your time and attention.
Okay, Matt's up. We're gonna skip back to electron microscopy.
All right, thanks. All right, yeah, sorry for the little bit of miscommunication there. All right, yeah, so I'm Matt Mecklenburg. I'm the managing director of the EICN. That's the Electron Imaging Center for Nanomachines. And we're going to talk about what electron microscopy can do and what it can do for you. Okay, so we have some fun facts here. So fact one, electron microscopes can see atoms. They're one of the few tools that can see atoms, especially in 3 dimensions. Fact two, transmission electron microscopes are used to measure transistors in your phone. Every semiconductor fab out there in the world will have a bank of electron microscopes, and they'll have people like me that sit there all day and just look at different cross-sections of transistors that come out of wafers. So you take a big 12-inch wafer that's coming off the assembly line, and they can actually pluck out individual transistors and say, hey, how big is this one? What is the size of this one compared to this one on the chip? And actually see how well they're doing. If there's a fault in the machinery, if there's a fault in the process of actually making these transistors, they can actually see the individual point and actually pull out that defect and actually do some post-mortem on the process. Fact three, transmission electron microscopes are the only tool that can directly image proteins and viruses at the atomic level. So like when you have proteins and viruses that cannot form crystalline structures, the only thing you can do is put them in a transmission electron microscope. And this is why places like NIH and other facilities that are interested in health, places like Amgen, will use electron microscopes extensively to image proteins and viruses. They actually have data banks of proteins and various other structures that they've identified that they use for the study of drug development. An example of an image of a virus is shown here up at the upper right corner, and you can see all the different spike proteins on top of this virus, and it's been colorized here. They're all actually not a particular color, but it's been colorized here to see the different features within that virus. Just below that, you can actually see a gold nanoparticle in the individual atoms, atomic columns inside of that gold nanoparticle. So those individual columns are separated by about 2 angstroms. The lattice spacings in most crystals are somewhere between 1 and 3 angstroms, so this is a good representation of this ordered little crystal here. Okay, fact 4, electron microscopes have electrons traveling close to the speed of light. So this is a really fun fact that the electrons in the electron microscopes are traveling somewhere between half the speed of light to 3/4 the speed of light, depending on the microscope. And so our old friend Albert Einstein gets to feel very proud that all the features of relativistic change are put into these microscopes every single day. So if you ever asked what the purpose of basic research is, well, the purpose of basic research can be that 100 years later, your crazy ideas actually go into a tool that helps understand transistors, and helps understand viruses every single day. Okay. Fact 5, electron microscopes use magnets 40,000 times more powerful than the Earth's magnetic field. We have these really big 2 Tesla magnets. And yes, Tesla actually refers to magnetic fields, nothing electric like in the car. 2 Tesla magnets that go inside of these microscopes that actually steer the electrons down. They go through, from an electron gun, they get sped up to about 3/4 the speed of light. They go through all these different lenses. They go through the sample. This quantum mechanical wave of the electrons actually travels through the sample, gets phase shifted, and then interferes to form an image, and that's what we look at. So this is another example of fundamental quantum mechanics coming in 100 years later and producing these wonderful tools that we use every single day. And then we actually have wavelengths that are about 250,000 times smaller than that of visible light. The wavelengths inside an electron microscope are about 2 picometers in size. Optical microscopes are wavelength limited, so they can actually see the size of things that are on the order of the wavelength, so about 500 nanometers, or a little bit better if you have super resolution. But electron microscopes have wavelengths that are about 2 picometers. Can we see things at the 2 picometer scale? Absolutely not. Okay, I wish we could. We cannot in any way, in any form, see anything at the 2 picometer scale. These kind of images here are roughly about the state of the art, but somewhere between 50 picometers and 200 picometers, and we're limited to that scale because electron microscopes have terrible aberrations in their lenses, okay? Terrible aberrations. If an electron microscope was a person, it would be legally blind, okay? Just absolutely terrible aberrations, and we use tiny little aberration correctors to make it a little bit better, but because we cannot, in optical things, you can machine down to 1/10 the wavelength and make it basically perfect. We cannot machine down to 1/10 of a picometer in order to actually see down there and actually get the aberrations free and work at that scale. So, yeah, electron microscopes can do all kinds of cool things. Here's another example of a virus. This is a little capsid that's absolutely enormous. This capsid's about 120 nanometers across, and you can actually see cool details like this little portal where all the DNA comes in and out of or goes out of the capsid here. You can use these electron microscopes to see the individual walls of a carbon nanotube. So that's what we have at the upper right. And those walls are about 3.4 angstroms in size. And you can actually, even though this carbon is very, very sensitive to the beam, we can actually image at that level. Here's another example of a gold nanoparticle imaged at atomic scale. All of these things are possible with the different microscopes that we have. We can do cool different things, like we can look at the RNA editing machinery, so you can actually see the different components that make up this machine here, and you can actually identify the protein that's doing the editing. That's what's shown in magenta there. When we do tomography, we can actually see inside of these objects in 3 dimensions. These objects can't really form crystals that well, so this is the only technique to look at this at really high resolution. And in the capsid we showed earlier, we'd removed all the DNA, but you can actually see inside of this capsid here all these little noodles that are actually the DNA that's like trapped inside of that little virus. Here's an example from the semiconductor side where you can actually see the structure inside of a quantum well. So you can see the difference between the germanium quantum well layer and the 2 semiconductor layers on either end of that. You can actually see the interface nearly at atomic resolution. This particular image here is more limited by the sample than it is by the actual imaging conditions, but you can actually see that atomically sharp interface from the epitaxially grown layers really quite well. So I'd like to introduce our team. You might recognize one of these people, but then we have Ivo, Wang Hui, Hongzhou, our founding director, David Strugatsky, and Judy Su. And we have a really, really great team, and they're absolutely available to help for both assisted usage and if you want to get trained on any of the tools in any way that you want to understand them. And then I'd like to introduce the instruments that we have. So we have, in this picture, we have two pictured SEMs. One is coming soon, and we can show you where it's going to get installed. And then we have 5 TEMs shown here. The TEMs, basically all the data that I showed you so far from the transmission electron microscopes. The 5 TEMs here is missing one important one, which we'll talk about in a moment. But each one of these microscopes is at a different level of performance, and so you start at like the introductory microscope, the T12. You understand the basics of microscopy, and you move up to the more advanced tools, and then even to the really powerhouse tools that get you down to like atomic resolution on a routine scale. The one thing that's missing is our Krios G4. This is now in the building, and it is up and running. It doesn't look like much from the outside. It looks like a box. We can show you, and it has this beautiful blue light. But on the inside, it has all these kind of cool tools. Now, what's really, really interesting about this microscope is that this G4 in this configuration is the first one in academia in the United States. There are a couple at, there's one at a national lab, and there's a couple at pharma. But basically, this microscope has been designed to build on the resolution revolution that's going on in structural biology. And it is designed to do really, really high throughput, real-time structure determination of these viruses and proteins that we're talking about that are so fundamentally important to human health. And so it's got all kinds of tools that I can go into associated with that make it this really, really, really powerful tool. And we're really excited to work with people to basically output as much as we can, learning about both human health and the huge expanse of proteins and other structures that are in the biological space that don't get examined because human health takes such a huge priority. Any questions? No questions? Did I talk fast enough? Okay. All right. Cool.
Okay, thanks, Matt. Okay, so we'll jump ahead now up one level. So Wilson is going to come up next and talk to us about the UCLA Nanolab, which is our clean room facility.
All right. Hi, everybody. My name is Wilson Lin. I'm the technical director for UCLA Nanolab. It's hard to follow Matt, but we work closely together. He had tons of beautiful images with the lab that I'm making devices for them. So what we do, we can read this, but basically we're making chips or devices for your daily applications. All the way from your cell phone, computers, your car, biosensors, drug delivery systems, even artificial eyeballs. We make all kinds of devices in our lab. So you have any crazy idea you want to make, come to us, we can help you make them. All the devices were made in the clean room. For some of you who don't know what a clean room is, imagine in a 1 cubic food volume like this. Classroom 100, you have about 100 particles at 0.5 micron size in this space. In the air in this room, we will have about 1 million particles that size. So the clean room is extremely clean. So when you enter the clean room, you have a gown like this with your clean room gown, your face mask, gloves, shoe cover to protect particles from contaminating your wafers. Also, we have one of the most state-of-the-art clean room filtration systems. All the air will push from top to the bottom, push all the particles to the bottom of the floor, circulate air back to the filter for you to provide a very clean environment for your devices. All right, so this is like 1 minute introduction was a clean with this. These are my team, my staff. We have total 10 staff on our team who have extensive experience that can help you either train you on the tool or make you make your devices. We have over 100 equipment in our lab, and we like to break the tool into 3 categories: lithography, deposition, and etching. Internal lithography, we have e-beam lithography, who can write a sub-micron devices. We have optical lithography, can write feature size about 1 micron and above. We recently just purchased 2 brand new direct writer tools. One can write about 300 nanometer system, and the other one can write about 600 nanometer feature size. We also have a wide range of imaging systems, SCM, FIPS, and we also purchased a brand new top-of-the-line SCM tool just recently. We work very close with Matt about the imaging system that we sort of use each other to sort of complement each other. In terms of deposition, we can deposit metals, insulating films, dielectric, polymers, and the technology that we use can range from e-beam evaporator, spotting tools, chemical vapor deposition, PECVD or LPCVDs. We also can do spin-down photoresist, any kind of creative way that you can deposit the film on your substrate. We know how to do it, so talk to us. We can help you with deposition. Once you have your film deposit, then once you have a lithography to pattern your features, next step is etching your process. There are 2 ways to do it. Typically, people will use a plasma etching, which is a vapor phase etching system. And we also have a wide range of wet chemistry allow you to pattern your devices. Once this is done, then we'll take an image to look at how the wafer looks like, what's the cross-section on your device, and how they look. And we also have a lot of measuring systems that measure CVIV, electrical property of your device. If you're interested to learn more about our lab and what do we do and want to learn how to make devices, we offer a wide range of classes in our lab. If you're UCLA students, we have a quality fabrication class. We make CMOS, MEMS, and other devices. We typically have over 150 undergraduate students go through our program. If you're a graduate student, we partner with physics department who also learn the similar clean room experience as well. If you are non-UCLA students, we also offer summer programs, which allow students to come in to spend about 2 weeks with us, and we will show you what the clean room environment looks like and how to make device within our lab. All right. Good. All right. If you guys, I know it's a very short version of what clean room does. If you have time, come down here at 2 o'clock. Myself and some of my staff will be there to show you the lab, to talk about more detail, what we can do and how can we support your research and your fabrication process. All right, thank you.
Okay, thanks, Wilson. So that's our transition from measuring to making in a very, very clean environment. Now I'm going to turn it over to making things in an entirely different environment with a little bit of shift towards some automation and high throughput with Mike Lake to talk about BioPacific.
Yeah, it brings up a great point of what is clean, right? Having a packed room with tons of stuff in it or just having a bare space with nothing in it, right? I'm not sure cells are really, really dirty or exceptionally clean, right? We'll have to see. That's why they do good chemistry. Anyhow, so that is actually part of what the Living Biofoundry is about. We are a robotic, automated facility, basically, I mean, at the basic level, trying to get people from spending their time actually pipetting after they've had this great idea to actually being able to have robots that can implement that pipetting, but also bigger to be able to scale it up so that we can begin to think about biology in a 100 experiment at a time way to solve the biological problems rather than asking, did our assay work this week? Right? That's what we would try to do. And again, it takes longer to get a robust assay working, but when you have more and more and more of your component automated, you can start doing your science faster. So the real purpose of our facility is to actually accelerate the rate of scientific advance, not just have you not do as much pipetting, if that makes sense. Both are good. So with that, I also, that's the Living Biofoundry. I should step back for a second. I'm also representing the rest of BioPacific, where we're housed. So we are the production side on the Living Biofoundry to make monomers that actually have a further life that then go into polymers that are made by our collaborators at UC Santa Barbara. And so part of the schema that we operate under for the NSF is to basically try to create materials that would not be accessible if you did not start with things that could only be made in a cell, and then move them to a material that can only turn from a goop in a cell, some liquid you extract, right, into an actual hardened 3D material when you send it to polymer chemists like up at Santa Barbara. So we're going to talk about that nexus across all 5 of these facilities, starting with the automation design principles that I talked about, meaning we're trying to get higher throughput, more cycles, more shots on goal, and we're trying to take advantage of the idea that we can practice the science fast enough to also design the experiments to generate inference, inform, and initiate a new cycle that's even more productive than the last time we did it. The difference is when you do it the second time, the robots are still ready and waiting. The first turn of the cycle is much harder. So with that, this is the Living Biofoundry. Let me go ahead and, this is actually our facility upstairs. It's a Thermo Fisher lab automation system with a whole bunch of extra bells and whistles we've customized and added. If I start this video over here, this is what it looks like, taking a plate out of an incubator, moving it around. And while this plays, I'll tell you what we really are all about is, like we said, the DBTL cycles, what I've been talking about, it's design, build, test, and learn, right? And I would say one of the most important things we are trying to work with our users is trying to get away from that linear model of research, where we set out to accomplish this goal, we do these experiments, we get our conclusion, we publish a paper, and then it stops. Oh, thanks. We want to return that to basically say, okay, we finished. Now let's input what we learned from that last cycle, repeat. And so we want your project, we want you to have the one extra thought. What are you going to do if you get 500 answers or 1,000 answers or whatever it is we're capable of delivering to you that then allows us to start the next stage of the experiment and advance and accelerate the development of materials as opposed to fixating on, like I said, did you get 500 measurements, get some good data sets, and get a publication, right? And publications are great. You get them at each stage, right? You can publish at each turn of the cycle. That's fine. But acceleration is a different concept. So with that, we also have all these standard processes like PCR, automated liquid handling, plate readers, the standard stuff every biology lab already has and knows, but we're trying to integrate it into this more automated framework. So with that, I'd also add, adding on to what Adam said, that's the commercial stuff. It's all operating and come down. We can talk about it. And what I would say is, as you all are probably familiar with in the biology side, a standard set of equipment can accomplish an amazingly wide amount of goals in the biology space, right? This same robotics equipment is used for high-throughput drug screening. It is used for COVID testing. It is also used for biomanufacturing. Right? So we have incredible capability. The key is, can we thread everything you want through an automated platform so your entire assay becomes automated? To wit, one of the gaps we identified was that we had an electroporator we bought, which is the best in class, twin wave, works on yeast bacteria and mammalian cells, but was not fully automated, right? And the fully automated version that only works on stem cells is $150,000. So what we did is we actually built our own robot to press the button and a robot to raise and lower the actuator with an incoming freshman, by the way, in bioengineering, as well as a fourth-year physics student. We're also coding all the electronics. And that's our control electronics. I didn't show you the gory code, but we're also writing code with REST API. So it's actually going to be run out of our Thermo Fisher original suite, just like all the other professional devices in our line. So that's the idea. And I would just say to all of you who are interested in coming to do projects with us, we will take whatever idea you want to feed it into this infrastructure and maximize the impact of it. And if there's some specific gap that's just killing this amazing thing from happening, we'll try to build through that gap as well. I think that's what we mean when we say we're not just a place where you can come and do some push button stuff. So with that in mind, let me tell you one thing we're actually doing on the subject matter. And this is, for example, we do a lot of biopathway work. We make lots of small molecules. We are by no means limited to proteins, right? But as a protein example, this is a great example of what like a biofiber or a biomaterial of the future might look like. These are called SAM synthetic fibers. Also, we are expanding all the time our remote capabilities and our remote access. This is a collaboration we have with the University of Washington in St. Louis. These researchers are actually remotely operating our tools as well as collaborating and sending resources back and forth to accomplish this goal in our facility. And believe me, it'd be a lot easier if they could just drive over it, like some of you that are local. But what they're doing is they're combining three super performers in the biomaterial world into one fiber that can outcompete cotton and nylon in some of the different categories, right? And that's because they're combining a silk fibroin, we've all heard of that. They're combining an amyloid plaque from Alzheimer's, which is very, very tough, with also muscle foot protein, right? The stuff that gets stuck on boats, right? Those are three huge performers. Put them all together, and then that's just the starting point. Because then imagine you're going to take that as your starting point and begin to engineer it and advance it through multiple cycles. Your later won't be any one of these three exact proteins. It will be the optimized phase space where you figure out how to get something softer and more flexible or something firmer, et cetera, beat cotton and nylon in whatever your application is. That's the kind of idea for a higher tech framework for biomaterials development. So let me move on from that to where we talk about the automated synthesis. So this is where we get to automated synthesis in chemistry, right? We have at UC Santa Barbara a ChemSpeed, which is basically an automated suite that does all of the things you'd find on a bench in a synthetic chemistry lab. Right? So analogous to what we were talking about with the robots, but we're not going to see them today, so we're not going to go into as much detail. But it also is a glove box condition. You can have low oxygen environment. You can do things that we'll never be able to do. They also have a flow chemistry reactor, and they also have a peptide synthesizer, right? So you can imagine the flexibility of what you can actually produce and where you can make new monomers, new peptides, for example, that can go into a new monomer for a peptide synthesizer that then goes in to make a new non-natural amino acid into an actual structure or into a material, right? There you go. So there's a chain from biology straight into that. We also have additive manufacturing, and here you can see we actually have this, one of the first collaborations with carbon to actually use the CLIP process, which actually generates things at the liquid-air interface, generates polymerization at the liquid-air interface, and it's 200 times faster potentially for making materials. We also have control. If you see those two dyes, that's Psi3 and Psi4, but what that is, think YFP, right? Your favorite protein. Right? They have a printer that can make sure your stuff ends up here and your other stuff ends up here, and it's structured into a polymer that can have the properties you want, hard, soft, flexible, what have you. Right? So really amazing things. This is, again, what do you do when you take something amazing and make it in biology? You got to throw it into something that's structural and useful. So we have both shape as well as chemistry working in terms of the collaboration. High-throughput characterization, as you guys have already heard a little bit, is another thing. What do you, again, design, build, test, right? Just like electron, great examples Matt gave of how important electron microscopes are for actually characterizing things. We similarly have an unbelievable best-in-class X-ray wax and sax line that, if you can see here on the bottom, is actually competitive with data you get from a synchrotron. It's not better than a synchrotron, but you don't have months of delay to set up. They run it fast and quickly and easily. They developed a higher brilliance source than any other labs, and that was the basis of creating a custom tool that is now widely available and under very high usage, but also very, very easy to use. You can literally mail them samples, get your data back, or go show up, get trained, and use it for yourself. So it's really, really wonderful facility. You've already heard about electron microscopy. You haven't heard that they have another specialized microscope in micro-ED that actually does microcrystal electron diffraction. So this is another example where you can look at polymers, you can look at proteins, you can look at drug substances, right? And basically, if you see on the left, yes, you have scattering. We're making diffraction patterns now, not images. So a little different than TEM for those who are not familiar with the differences. And on the bottom, what you can see is you have a tiny little crystal that's actually on your TEM grid, right? That's TEM grid, little crystal. And what you get is a diffraction pattern for it. And now all of a sudden, you can actually solve the structure of that. And as we get to, again, you guys saw the examples of biology of how far it's come. Imagine when we can do that with polymers, which again are never going to have a perfect repeat crystal structure, right? Biopolymers can benefit from this tremendously, especially as it really gets off its feet. And then the last is mechanical characterization. We have an optical indenter that can give you physical characterization in 96-well format, right? We can do both viscoelastic characterization, static characterization, right? So you make the newest hydrogel that's super exciting. You want to know for sure your hydrogel is exactly this with this kind of conformity. Great, you can make 100 examples and test it all for quality control. You can also make a gradient of it and take the one that you find is the exact one that you're, or make a gradient of them, verify what they are, and then grow your cells in them and see what the effect is. So really, really great baseline for when you want to do biomechanics. On the bottom is microrheology. So that's anything that's liquid, right? They say anything from water to honey, right, is basically what you can measure with microrheology, and again, extract its elastic properties, right? So this is very helpful for 3D bioinks. 3D bioinks often are limited by the viscosity as they get out of printers, right? Here you could make 10 or 15 very small quantities of your test bioink, see which one actually is going to conform and be most likely to print, and then scale that up. Rheology tools usually require 3 milliliters by comparison. So with that, I would just say thank you all for listening, and I'll be available for any questions. We're downstairs, and we'll have a bunch of robots running. I always say it's like model trains. So come on down, and while the trains run, hopefully we can talk about all your exciting projects.
Okay, and now all the way up onto the 6th floor is our signature facility for automation, drug discovery, and all things that Robert has been building here for the last 20 some odd years. So we're excited to wrap up with Robert.
Thank you. Okay, so this is a little bit of a different facility from everyone else because we have a unique focus. Our focus is making drugs, developing drugs, finding drugs, and getting those eventually to the patient. Now, how do we do that? We start with you. This means we start with your ideas, we start with your vision, we start with your concerns that you bring to us, the disease that you care about, and then we take that forward and we wrap that into a discovery, into a journey that we take you on all the way into the clinic. So I'm gonna step you through the individual bits and pieces here. But before we get there, let's talk a little bit about what we aim to do as a facility. What we do want to do here is we want to bring you top-tier infrastructure, expertise, access to small molecule functional genomics libraries. We want to transform scientific discoveries into medicines. And we do this for the patients in California and worldwide. The other thing that we really want to do here is we want to teach you. We want to teach you how to discover drugs. We want to equip you with the skills that you need to be successful out there in the pharma world, maybe building your own enterprise, building your own company. This is where it all starts if you want to be within drug discovery, biomedical research enterprise. This is what we like to do. Okay, how do we do that? We focus on high-throughput screening. It's a productivity tool and has many applications, but what is it? It's really the execution of the same assay at a scale that's industrial, more than 10,000 times per day. Now, let that sink in. More than 10,000 times per day, with reliability, with basically no fails. So, what can you do with that? Drug discovery. So we talked about that, we belabored that. We can screen all sorts of libraries, targeted drug-like diverse libraries, but there's a lot of really interesting applications that are maybe a little bit more up your alley if you're a basic researcher. One of them is functional genomics. You can use CRISPR, cDNA, SI, all of the tools that you're used to. You can do this in high throughput. Or chemical genomics, where you're not modulating the genome, but you are actually changing blocking key pathways, signal transduction, etc., using small molecules. It's a very fast, maybe a little quick and dirty approach to discovery, but it's uniquely powerful. And then, of course, there's materials discovery, characterization, toxicology, etc. But today, I just want to focus on drug discovery, genomics, and so on and so forth. So how do we give you access to high-throughput screening? We give you access to a very unique environment that's roboticized. And you see this here in the middle, you see a robotic arm that travels up and down. No, I wonder if we have something like a laser pointer. We do. So that travels up and down a little rail here and connects all of these things that we call peripherals. This can be incubators, this can be automated microscopes, plate readers, washers, dispensers, liquid handlers, you name it. We're covering everything that you might do by hand normally in your lab with automation. What this enables is, and what this gives access to is, the ability to run your experiments 24/7, 365 without anyone having to be in the lab. Your productivity, and I'll show that in a moment, is super enhanced. Okay. The other thing that this gives you access to is really the ability to screen our small molecule libraries. We have about 330,000 very well curated small molecules. And just as a comparison, I think Gilead has about 750,000. They're getting slightly nervous because we're approaching their size. There's a little bit of overlap, so we are up there mid-size pharma level. It's very unusual to have some resources like these. But what really sets us apart from everyone else is functional genomics. Genome-wide, SI, now here's a lot of stuff that's cut off. There's CRISPR missing, there's cDNA missing. There's a lot of other things that I'd love to tell you more about if you come and visit me on the 6th floor. Just to show you, give you a graphical impression of what this means to go from manual to high throughput. Just brought you what one user did with us. They were screening 10 IC50s, dose response experiments in triplicate a week, and they went from that by hand per person to 250 in an afternoon. That's the difference. That's the difference from manual, automated. That's tens of thousands of data points in an afternoon without you even having to stand there. So, at the end, what comes out of that? What comes out of that are molecules like 3515, which is the first small molecule that is UCLA homegrown, that made it into phase 1 clinical trials for oncology. It's gotten finally orphan drug status. It's now moving into phase 1. I have heard that it now passed phase 1 with flying colors, which is exciting for us. But this is really what comes out of our research when it's successful. Another success story is DNP19, which is an antibody that you can load up with PET isotopes or with drugs or. It binds to LRC15, and that's a TGF-beta-driven biomarker. And you find this on the surface of very aggressive cancers. And you see how this particular antibody here, you see this in yellow, gets internalized when you have a cell and when it binds to it. That's really important for such a drug because that means that this gets taken up in the tumor and doesn't come out anymore. What this also means is that it actually is able to knock down the tumor growth. You see this here in a mouse model of an osteosarcoma, where after one treatment at 30 megabecquerel, basically the tumor is completely ablated. It doesn't come back. 160 days later, it's still gone, which is an excellent result. Also, of course, importantly, you don't find this drug anywhere else in the body. This is, again, a mouse model here. You see osteosarcomas, here HU09, SAO2, U118, different types of tumors. It gets taken up really, really well. But this is what it looks like when research in my book is successful. Okay, so I'm going to stop here and see if you have any questions. if you would like to get in touch with me, my email address is down here. And yeah, I'm looking forward to seeing you upstairs on the 6th floor. Thank you.
Okay, so thanks to my colleagues for the warp speed intros into all the things that they've built and do every day. Just as a quick reminder, we're going to wrap up here and we're going to sort of unleash you all to the labs. We've done it before where we have sign-up sheets and it tends to be more trouble than it's worth to try to coordinate where everyone's supposed to be going. But just as a quick reminder, for those of you who've not, or who've been here, those who have not been here, there are elevators on both sides of the building, right? We're going to be headed down to the basement level if you are looking for the characterization labs. And so there are specific rooms that you'll see highlighted. I believe the last slide shows the exact room, but there are these posters on the wall, so you'll sort of see where to go. The A level, as a reminder, is going to be the entire A level is one of our clean room facilities. The second floor, room 2145, is where you'll find the living biofoundry, where Mike and his team will be. And then on the 6th floor, above us up on this side, 6310, is where the MSSR facility is. As you're walking around, I'll just also point out, the technology centers occupy about a third of this building. You may see some other labs. You're like, oh, what's going on in that lab? And so we have, partners that are inside of the building as well. Some may have their door open. I think I see Baolai here, who is the director of the Integrated Nanomaterials Lab. So for folks interested in compound semiconductor epitaxy, deposition, growth, characterization of those materials, Baolai will, I think, also have your door open. Is that correct? Yeah, Baolai will be available if you want to stop by. This is next to the Living Biofoundry. On the 2nd floor, you may also walk past the Crump Preclinical Imaging Center. They are not part of the CNSI technology platform, but they're in the building and they're collaborators of ours. They do micro-CT and positron emission tomographies for small animal imaging. So if you walk by and see that, there's a poster there and the contact information is available. And then if anyone's interested in low-dimensional materials, magnons, phonons, different optical and mechanical properties, we do have a brand new Brilliant scattering facility downstairs. The door is not going to be open, but if you're particularly interested to see it, you can ask me because I have the key to that room and we can show it to you. But by all means, it can certainly connect you with the folks that run those facilities as well. And so a lot of what we do here, obviously, as you see, is bringing advanced technology to bear and democratizing access to it, right? And so what we're really excited is that you're all here and interested, and we look forward to seeing you in the labs over the next couple hours. And then, of course, at 4 o'clock, the lab directors will make their way upstairs, and then we'll have an opportunity to have a little bit, less formal over some snacks and a drink, and wrap up the day. So again, thank you all for coming. If you have questions, you can certainly contact the labs directly. All of this information is on our website. You can email tech centers at CNSI. That goes to a handful of us that check that. That QR code will also take you to the right place if you want to get to where you need to go to find more information. But we're always here. We're open 24/7. We don't necessarily all answer our phones 24/7. I probably do more than I should. But nonetheless, now you all know where to find us. You know who the contact people are. And thank you again all for coming. And so with that, I will turn you all loose. We will head to the labs and look forward to having some good conversations. So thanks again for coming.